basically what I cover in the lecture is the things that I could test you on. And there's a lot of material in the chapters. It's kind of too much material in the chapters, but it's good to read all of that stuff and put it in your mind. You know, the best formula for doing well in this class is to read the chapter ahead of time that I'm talking about. And even if you don't understand some of what's going on there, just read through it. Just force yourself to, okay, I didn't get that, on to the next paragraph. You know, just put it all into your mind. And then when I talk about it in lecture, some of those connections will be made in your mind. It's like, oh, now I understand what that part was about, whatever. And then, you know, before the exam, you can study again, and then you'll pull it all together. But the material that's in the lecture is the material most likely to be tested. And so then it's good that we have the tape going too, because if you're unclear about what that was, you can just go click on it in Moodle and you can see, you know, most of what we discussed. And we'll see how it goes. You know, I grade on a curve, so if people are not getting it, we'll try to make some adjustments so that everybody gets it. It's my objective to make sure that you're all well trained doing this. I'm not interested in leaving people behind. But on the other hand, you know, we have a lot of material to cover. I think it makes sense to cover the complete human body, right? I mean, you know, we are in a unique situation to tie together some other biological information, some other coursework that you've had, kind of give you a whole picture of what's going on in the human being. So I think we should take advantage of that and cover all this material. So it's a pretty rapid pace, though. You'll see. Week after week, we're going through different organ systems and that kind of thing. So there's lots to do. Okay, so let's see. Start out. So in talking about the bone marrow, the cells that are in the bone marrow it's still working.
That's on.
Yeah. Those are stem cells, right? So remember last class I drew up here stem cells, and then they give rise to progenitor cells, and these are pluripotent cells, and they'll give rise to a variety of blood cell types. So there's only one stem cell. So that's the interesting thing. So if you've isolated the necessary stem cell, you could repopulate the bone marrow with that stem cell, and all of the blood cell types are going to be available. That's pretty impressive. So it shows that one stem cell type has the capability, it's pluripotent, has the capability of giving rise to all of the lineages. So they're all related to each other. But what's different about them is that they're in these different niches within the bone marrow. And I was drawing a culture dish and showing colony forming, and saying little clumps of cells. You know, clumps tend to be of one cell type, and they'll give rise to, you know, only erythrocytes, and so then that would be erythropoiesis. Or if they're giving rise to just platelets, it's thrombopoiesis or granulopoiesis, monocytopoiesis. You know, each one of these cell types are going to be budding off and giving rise to cells. And then I showed from the biotech companies, remember there was this pipeline, and I talked about the pipeline of some biotech companies, and some of the factors that are involved in maturing blood cell types are their products. So that's, if you went to work for, you know, somebody like Genentech locally, some of the things in the pipeline are growth factors and cytokines required to mature blood cell types. So, for example, GCSF, granulocyte stimulating factor, GM-CSF, granulocyte macrophage colony stimulating factor. These are products. So not, you know, they're normally produced in the body, but when we make the observation that a patient is low on a certain level, there's sometimes a therapeutic benefit to being able to stimulate those cells to produce more. And so then you can try to normalize the blood by adding some of these factors. And sometimes, you know, this has to do with chemotherapy, for example. You know, you've given a treatment, so you're trying to eradicate cancer in the body, but in the process, you're also poisoning some of the cells, and you end up getting a reduction, for example, in erythrocytes. So if you wanted to stimulate erythrocyte production, you would also enhance the amount of hemoglobin being made, and that's where EPO for erythropoietin is a blockbuster drug, and then there was this company Amgen that made billions of dollars just on EPO, and it's one of the best stories yet on a really useful substance that was identified and then offered as a product and has a lot of benefit in the clinic. Okay, so what I think is really interesting about the bone marrow, though, is that it's solid tissue. We've made that transition away from the cells in suspension into tissue. And every time you have a tissue, you have cells adhering to each other, so that's one part of it. But then you tend to have two cell types too. The specialized cell, sometimes called a parenchymal cell, specialized cell that has this function, and then some sort of support cell. So some sort of stromal cells that are around those. And the stromal cells are producing proteins, and a lot of those proteins are extracellular matrix proteins, ECM. And so we identified some of those proteins on the board. We talked about collagen and laminin and fibronectin, right? Remember that? So those proteins are an important part of the signal, too. I think it's very interesting that these stem cell progenitors that exist in the adult bone marrow actually start out at other locations during embryogenesis. And a certain part, they're in the liver, for example. And then, with the mature adult, once there are good bone cavities to populate, they kind of migrate there and end up as that's the main area where blood progenitors are produced. So that's sort of interesting, right? Because you might have a sort of a static idea in your mind that things are built in a very conservative way, but in fact in the body cells are flowing around to different locations, and as you make the transition from a developing fetus into an adult, like some of these cells migrate to new locations, take up residency, and then do their thing there. So that shows how dynamic the whole process is. It's pretty amazing. So just look at, let's look at this one for a second. This is where the blood cell types come from. Figure 13.2 in the book. And this is worth looking at and knowing. So basically that there's one stem cell type that gives rise to all of the blood, and that there's two pathways that it can go in. It can either go myeloid or else lymphoid. So most of the blood cell types are myeloid, so that's giving rise to a myeloid stem cell, and then you have colony stimulatory factors that give rise, when stimulated with these specific factors, give rise to a lineage. So that could be erythropoiesis, thrombopoiesis, leukopoiesis. So it starts out always with a progenitor cell, and then the first sort of immature cell is something with the word blast at the end. We were saying that a site, like an erythrocyte, erythrocell, that's the mature cell. And when you see the word blast, that means it's typically an immature cell. So rather than having you guys remember that progenitor goes to erythroblast goes to a polychromophilic erythroblast, I'm not going to ask you that per se, but I want a deeper understanding from you guys about what that is. That you should always know that the blasts are the immature cell, that some of these words, the basophilic or polychromatophilic, orthochromophilic, those are all, it's funny because those are descriptions straight from histology, right? So people were trying to figure out what's going on with the blood. They stained them with some different types of stains, and we talked about positive binding anion or cation binding stains, and so whether they're binding or not has to do with their name. If both of them bind, you got a polychromophilic. If one of them stops binding, then it stops on one side, so it's an orthochromophilic. But you don't need to necessarily know that these are following that. But you need to know that those names have to do with their uptake of stains, and so it relates very directly to histology, and they were named after what histologists were seeing. Of course, one of the most important steps is kicking out the nucleus. So once the nucleus is ejected from these earlier precursor cells, then you've got something that looks much more like an erythrocyte. And I think it's kind of cool that the reticulocyte is the thing just before the erythrocyte. It also has the nucleus kicked out, but it's still making hemoglobin. And because of that, when they stained with a certain crystalline blue stain, they were able to see some translation going on. Messenger RNA was being translated into hemoglobin, new protein, and so you get a blue stain towards the center of the cell. So that allowed them to differentiate a reticulocyte versus a erythrocyte. Okay, on thrombopoiesis, again, there's some sort of progenitor. The final cell is a megakaryocyte, and then there's some other steps, and the blast is the earliest step, right? So that's the kind of thing you need to know. And have the concept that these are very large cells, and they're just budding off little pieces of protoplasm, and that's what a platelet is. That's why platelets are so small. They're smaller than the red blood cells. They're just these little cell fragments, and they're crucial for blood clotting. And then leukopoiesis, again, start out with a progenitor cell, and you have two different types of blasts, myeloblast, monoblast. What you're going to be making here are these granular cells versus the agranular pathway, right? So all of these guys, the agranular, these guys are granular. So these are granulocytes, but they separate early on into these three different types. And the main one is the neutrophilics. So the neutrophils are a big category, and basophils and eosinophils are much less common, but they're involved with some specific responses. It's worth knowing a little bit about what those responses are, and we discussed that some already. And then a monocyte, is an agranular cell, but it matures into a macrophage. So that's worth knowing, that this is going to mature into a macrophage. If there's a slide and there's some of these blast cells identified, are we talking blood or bone marrow? Typically bone marrow, right? That would be one way to identify. If I'm showing a picture, and let's say I say, well, there's erythroblasts. These are erythroblasts in this section. Then the first thing you should think is, we're looking at blood cells, but the blood cells are still in the marrow. They're not yet circulating. Because sometimes the sections of marrow are kind of loose. Do you notice that? So you just see a bunch of cells, and so it doesn't look that much different than blood. It could be a little bit denser, unusually dense, but then there's going to be these premature, more immature types of blood cell types in there. And then, of course, a lymphoid line is completely different than the myeloid line. So, on this chart, everything's myeloid, which means blood, right? And then on this side, you've got lymphoid, which is a different system, and you've got T lymphocytes and B lymphocytes. They start out as T lymphoblasts and B lymphoblasts, and they mature down. There's no intermediates to know other than you go from blast to site. So it's good to have that idea, and then be able to identify the difference between blood and bone marrow. And we'll show some pictures where there's a disease, just like you guys did your presentations. And so remember, if you don't know the answer, please rationalize through things. Tell me as much as you can about what's going on. Professor, I don't know, I see that there's red blood cells there, erythrocytes, and then there's some other cells type there, okay? I don't know what that is. Well, is it granular or agranular? Remember we went through that last week. Pretty easy to say. And then if it's granular, it's got these spots, these granules in it, then it has to be one of these three, and neutrophils are so much more common than the other things that, you know, first guess is neutrophil. So it simplifies out pretty quickly. Okay, so let's see. Yeah, on figure 8. Well, that's the maturation of these guys. It is. This is a pretty clear cartoon. So we talk a lot about some kind of capillary. So in the circulation, there's, you know, veins and arteries. These are larger blood vessels, but we're dealing with microscopic tissue sections all the time. So that's why we're always talking about capillaries, and a synonym for capillary, microvasculature, microvessels. So don't be confused by that. And constantly, we were saying in biology, a bunch of synonymous words used to describe things, and you could think, oh, this is incredibly complicated, I don't understand what he's talking about. Or you could, you know, just try to list those words together and say, okay, you know, capillary is part of the microvasculature. These are just small vessels. And we're interested in those initially because we're always looking at microscopic tissue sections. So inside all tissue, there's going to be blood. It's going to be blood vessels. And we were saying that, well, all cells need to be close by blood. How close? How far away can you get from a vessel? We threw out that number. Remember it? How many cell layers can you be away from a blood vessel and survive? So like there's a blood cell right next to this capillary here, right? Here's a cell right next to it. But how about if you're, you know, can you be, how far away can you get? And we can put it in terms of layers, like how many cell layers? 5, 10, 15, 20? 5? It actually turns out to be a little bit more than that, maybe 10, maybe 10 to 20, somewhere in that range. Not more than that, though. So that's pretty amazing. And, you know, so you're still talking micrometers. You know, it's very little distance. So therefore, there's got to be blood vessels everywhere. And that's why I thought, you know, it's an awfully good idea to pay a lot of attention to the blood supply and understand what's going on there, because it allows everything else to live. You're going to see blood vessels in everything. Every time we look at a different organ system, there's going to be some, you know, part of a microvessel or whatever, and so that's supplying the blood. So when the blood is flowing, it's flowing through these tubes, and the tubes are made out of cells, right? Is everything in the body made out of cells? No. Right. What else is there?
There's bone.
Yeah, there's bone and cartilage, which is related to bone. Like cartilage is not calcified, but it's the same kind of setup. Hair. Yeah. Yeah, there's a bunch of things that are not exactly just packed cells. We're not just a bunch of cells. We are cells plus everything that the cells have made. Right? So they make the stroma are the types of cells, stromal cells are the cells that love to make extracellular matrix. They're pumping out material. They're making lots of collagen. There's so much collagen made that if you buy a piece of an animal from the supermarket and you put it in the oven, you get this juice. Remember we were saying, and then you know that hardens in the refrigerator, and well, that's a gelatin, right? And gelatin is denatured collagen. So I mean, any part of any animal cooked is going to release denatured collagen. That tells you that there's a lot of collagens present there. So we're made out of a lot of extracellular matrix material as well. But these vessels are tubes that are, you know, they try to draw the individual cells here. Each one of these is an endothelial cell, and that's what they're drawing here too. So even though it's a tube, it's not like a piece of rubber tubing, right? There's spaces in between tubes. And once there's a space, you can start going in between the cells, even without damaging them per se. You can just sort of push them aside, and the megakaryocyte can, inject some platelets into the blood supply just by hanging out on the outside of that. And then these leukocytes flowing in the blood. Here's a neutrophil. It's a neutrophil probably. And they can squirt through and end up in tissue. So there is exchange of cells from one location to another, because things are not so solid like we'd like to think we're made out of plastic or something, but we're not. We're these cells, and the cells are moving around, and they're maturing, and they're turning over, and they're changing locations, and they're one of the things that we've only started to appreciate is how flexible they are in terms of differentiation. That a pluripotent stem cell can give rise to so many different things, and the blood is a great example of that. So anyway, so we're all comfortable with this idea, right? The blood flow. That was real basic stuff. The next slice of marrow. So if I said this was blood cell types, here, identify the blood cell types in the slide. Well, you know that this is not soluble blood. You guys looked at blood, right? You see a lot of erythrocytes. You don't see individual erythrocytes, and things are kind of packed together. So then you know, okay, this is just blood. It's got to be marrow because it's packed in blood cells. So, and then because what they were trying to show here is that remember I was saying about the clumps. The cells clump in the marrow in different locations, and a clump of cells that are producing granulocytes, the process of granulopoiesis is going on, and so you've got this clump of that. And then where you have erythrocytes on the other side, this is erythropoiesis. So this is part of the bone marrow. So you guys would be able to identify that for me, differences between bone marrow and blood. I thought it was interesting that they said that neutrophils can increase in number just from a lot of physical activity. So somebody might not have a disease, but they could actually have a somewhat increased number of neutrophils, which would eventually go away a couple of days after you've worked that hard or whatever. If it's a longer-lived increase in neutrophils, probably some kind of chronic infection. So the body's trying to chase down the invading organism, and so you have an increased population of neutrophils. That's a simple type of phenomenon. Okay, one of the things, I'm not sure I discussed this a lot, but apoptosis, you guys have heard about that before? Apoptosis. Okay, apoptosis is programmed cell death. And at the point where we're talking about differentiation and genesis of these brand new cell types, erythropoiesis, granulopoiesis, generation of a lot more cells, suddenly we're talking about cell death. Why is that so important? And it's important because you need to get rid of some stuff in order to program cell death. In order for other things to form. Sometimes they talk about during embryogenesis that the digits in the hand are actually formed by apoptosis, so that our hands are one solid thing, and then cells in between the fingers die off or whatever, and you end up with these individual digits. So that's an easier way to remember that, oh yeah, apoptosis is this programmed cell death. But one of the reasons why that's so important is that if you have a cell and it necroses, it just dies by normal process, necrosis, you know, it's damage that somebody gets, you know, there's an injury, physical injury, you could have tissue necrosis. That just means that, you know, cells are ripped apart and everything goes flying. All the components go flying into the surrounding tissue, right? So there's going to be all sorts of molecules in the interstitial fluid that shouldn't be there. Cells are bursting open. So that's a necrosis situation. Apoptosis is a shriveling situation where the cell shrivels up and it stays intact, but it kind of becomes a lunch for the surrounding cells. So the surrounding cells, they'll engulf this and digest it and reuse a lot of these macromolecules. So apoptosis is actually a very friendly thing. It's, you know, it's the recycler. It's and it's because it's recycling, it stops inflammation. Because this process here, necrosis, this is big for inflammation. So you know when you have tissue damage that inflammation also is something that you're going to experience. It gets red, and it can get swollen, and you also get pus. What is pus? What's in pus? Okay, white blood cells. Good guess. I mean, that is part of what's in it. What else is in it? Andrea, what else is in pus? Yeah, so I mean, whatever was infecting it. So the white blood cells, the leukocytes are going to be chasing after things, but I mean, whatever they capture, or whatever, all that stuff, it's going to be in the pus. There'll be some bacteria in there. There'll be some white blood cells. What else is in there?
Serum?
Yeah. Yeah, sort of the liquid component of blood. Okay, there's a difference between serum and plasma, but what is that difference, by the way? Anybody remember that one?
Serum's got the all the growth factors and stuff like that. The actual elements. Plasma is just the 55%
Okay.
without the cells.
Okay. So how did they get all those growth factors? How did serum get that stuff?
From lice, red blood cells.
Yeah. Yeah. Exactly. Yeah. So there's if you allow the blood to clot and you get lysis of the cells, they're constant. They spew out a bunch of growth factors. And so then it's interesting, you know, it's just as an aside, in the lab, we use serum all the time to grow cells. So those are cells not only from blood, but they're broken open blood. So that's going to really send the signal to the surrounding cells, we're damaged, we're in a wound, we need to regenerate ASAP. So they're good growth factors. It's good for generating a lot of new tissue in a plastic dish. But on the other hand, if you say, well, this system represents something about normal cells, you're not on target with that. It's definitely going to be a wound. All the cells are responding to severe wounding. And it's funny, the debate's still going on like another decade later about how meaningful those results are. And I mean, I just think it always represents what's going on in the world. Here's a disease we didn't discuss. Thrombocytopenia. It's a bleeding disorder. You have a reduction in the circulating platelets. So if there weren't enough platelets around, you're not going to get a good blood clot. So then there's going to be more bleeding than normal. And so it shows up on people like little black and blue marks all over the place. So you see small purple spots. That's right. They're called thrombocytopenic purpura. Little black and blue spots all over because the patient is not sealing up any kind of damage. You know, even in everyday life, we get bumped and various things happen, and we don't bleed from that necessarily. But if you didn't have enough circulating platelets, that could be a problem. Okay, so that's it for what I wanted to say about the bone marrow. And now we're going into the epithelia. And this is really where the rubber hits the road, because epithelia is everywhere. And as I said, I work a lot on skin. So in the skin, the outer layer of the skin is an epithelia. It's called the epidermis. But it looks very similar to the other epithelia in other parts of the body. And so if we just draw a picture of a slice of tissue, what is the epithelial part of that? So this is some sort of slice of something. If this is the outside, and this is inside the body of the animal, then where is the epithelia? These are tissue layers number 1 and number 2. Yeah. So this is the epithelia here. And I started to talk about this last week where I said, well, the outside is actually anything that's facing stuff coming in to the body. So sure, it's the outside of the skin, but it's also the inside of your mouth, because that's facing the outside, basically, right? Still. And then you go all the way down the digestive tract. Well, there's lining to the stomach and lining of the small intestine, large intestine, and then inside the lungs and the trachea and the lungs themselves, there's a lining. All of that stuff is epithelia, because it's facing, it's facing the digestive tract, or facing the air that you're taking in, or facing something that's in a sense, on an outer surface. And then this number 2 is something, a category we could identify as stroma. But it's not the only word you could use. In the skin, the stroma is the dermis. And the dermis is the main cell type there, are fibroblasts. But it is a stroma. It's a supportive, provides a support function for epithelia. I think that this is kind of amazing. Where are there blood vessels? We were saying that you can't have a tissue without blood vessels. So, is there blood going through this whole thing? This whole assembly? You would think so, right? Is there? Yeah? Okay, so if we drew blood vessels, it would look like that. It's kind of going everywhere, right? No, it turns out it doesn't go everywhere. Which is really one of the amazing things about epithelia. It's like there's no blood vessels in any epithelia. It never goes there. It actually will hug along the border of the epithelia in kind of a very efficient array, kind of little feed-in points. So it keeps a nice, healthy blood supply just below the epithelia, but it never enters the epithelia. Isn't that strange? There's some other strange things too. Cancer. Most cancers are carcinomas. So the epithelia, is much more likely to develop cancer. Much less likely on a stroma. So that's kind of interesting. So when we're talking about this model, it seems kind of simple. You have epithelia, stroma, another word, a synonym. And so the mesenchymal. Describe something as being mesenchymal. Mesenchymal cells, stromal cells, mesenchyme, stroma. Same thing. You know, stroma is a word used more to emphasize the supportive nature of those cells. What are these cells producing that keep the epithelia alive? But if you're not thinking of it in that way, you just want to describe its own function, then mesenchyme. So epithelial mesenchyme is a very basic building block for all parts of the body. It's actually pointed out that there's only 4 types of tissue in the whole body, which is bizarre. There's epithelial. There's going to be the stroma, mesenchyme, and kind of this is a somewhat of a synonym too. It's connective tissue, which is, it's not exactly, these words are slightly different in their implications, but they refer to the same stuff. So epithelial mesenchyme, it's the type of tissue that mesenchyme is, or stroma is connective tissue. And then the 2 other things that we have are muscle and nerve. Isn't that amazing? So those building blocks, those 4 types of tissues make up the whole body. Kind of seems simple. And when you look at epithelia, the epithelia has a lot of similarity in different parts of the body. So you'll get to recognize what epithelial cells look like. They form sheets. So an epithelial sheet. If they're growing on a plate and you look at them, they might look something like this, somewhat cuboidal, and they're packed in next to each other. Sometimes you can see a little bit of space in between, something like that. So, and then from the top, it would just, it would look like, you'd see the individual cell. The cobblestone, they would sometimes describe that as cobblestone. You guys know what cobblestone is? It's those music roads, blocks of granite or whatever. So that's the way an epithelial sheet would look like. So, in the exam, if you see cells that are all, they look cobblestone-like and they're all connected to each other like that, that's going to be an epithelia. And you might not know what organ it is or whatever, but in chapter 4, they're discussing the epithelia in general, and so they give a couple of examples from various parts of the body, because they want you to have the concept of the epithelial sheet and what an epithelia is just generally. Epithelia is a covering, a lining. It protects. It also absorbs and secretes. So, the lining of the gut. You have to absorb the nutrients from the digestion of the food. And so that epithelia is facing the gut contents, all of that digested lunch that you just had. So that epithelia has got to absorb those nutrients. And then also, epithelia is lining any kind of duct or secretory gland. And so it's also the main tissue involved with secreting stuff. And so that makes it pretty interesting. It's also involved with sensory functions. Since it's on the outside, little hairs that are vibrating so you can hear and all that stuff. All that stuff is epithelia. So there are different types of epithelia cells, and so like what I just drew here, you could call these cuboidal. And yeah, they look, kind of geometric, like cubes. So that would be cuboidal. In the skin, as that layer moves up, they end up being these flattened cells, look like that. And in the skin, these are called squames. So that's the name for the skin cells that are flattened on the very end, but their shape, you can say the shape is squamous. So that's squamous, this is cuboidal. There are some cells that are even more elongated than this. And those guys might look like that. You know, really, they're not so chunky, they're not cube-like, so they're not cuboidal. So they're elongated like this, so we call them columnar. So those are the three types of shapes for epithelia. And you know, in a histology class, it's all visual. It's all about the picture. Dr. Spain and I were talking about the picture earlier today, and you know, saying that that's really what we want to make sure that the students are able to generate their own pictures and then able to interpret pictures. And there's a lot of knowledge behind understanding what's in the picture. So the shapes of the cells, even if you don't know the name of the type of cell, if I give you a tissue and you don't know that it's from a digestive tract or whatever, you can still comment that if it's flattened, if it looks like an epithelia but it's flattened, it could be a squamous epithelia from somewhere in the body. Or it could be cuboidal epithelia. Or it could be columnar if they look elongated like this. The other thing about epithelia is that there's got to be a good detector here to understand what the direction is of the cell, right? Because we were saying that this is the outside world, and this is the inside of your body. Then these cells are lining up. They know that there's a they can detect that, and they call that polarity. You know, in electricity, the polarity is whether it's plus or minus or whatever. But with cells, polarity is they understand the difference between the inside side of the body versus the outside side. And so what's this side called? The one that faces the outside. Per cell, each cell knows this. And they line up. They say, oh, you know, I recognize this is the inside, that's the outside, that way. There's a word for that. It's a biological word. You've heard it before. Yeah, exactly. And then what's the other side? Yeah, base. So just to distinguish from basal from apical, these cells have a distinct polarity. How about the stuff on the side? So that's, you know, facing outward and facing inside. But just, you know, the cell also knows what the surface is that's going to be next to its neighbor cell, and what's that one called? Apical, basal, and what? Medial. Yes. So it's better to guess than not see. But lateral makes sense, right? Oh yeah, it says I got that. Okay. So you know these terms. Yeah, I mean, it's nice that we're connecting all of biology together in histology. This encroachment from molecular biology and biochemistry and anatomy, physiology, and all this stuff. And you know, I want to foster that kind of depth of understanding. But still, you know, for histology, it's really all about the picture and being able to interpret what you're seeing in a slide. So that's why I'm giving you these terms. These might seem technical, but if you're looking at a picture and I ask you to describe